Welcome to Data in Biotech, a podcast from CorrDyn where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. This week, we're excited to be joined by Tommy Tang, Director of Computational Biology at Immunitas Therapeutics, a single-cell, genomics-based therapeutics company focused on immuno-oncology. During this interview, our host Ross Katz speaks with Tommy on the challenges and importance of data analysis in the field of computational biology, how to select the appropriate machine learning models to solve specific problems, what it takes to build an internal culture that encourages shared learning, and why he is motivated to build a community around computational biology. Here we go.
Tommy Tang, welcome to the Data in Biotech podcast.
Thanks for having me.
So just to get us started in a minute or two, would you mind just telling me about your career to date?
Of course, yeah. I came to the States in 2008 to pursue my PhD at University of Florida, majoring in genetics and genomics. But I was trained in a wet molecular biology lab, cancer biology. So I did a lot of experiments, pipetting, cell culture, all this stuff. So 4 years into my PhD, I was forced to learn computational biology by myself because my advisor at the time asked me to analyze this public available chip sequencing data. And I couldn't open it with Excel. It crashed. So I realized I had to actually learn some computational skills. That's why I started to learn Unix, Python, and R just by watching like online classes, reading books, Google a lot, and it's a lot of work. And then 2 years later, like after my PhD, I joined MD Anderson Cancer Center to pursue a full computational biology postdoc. At that time, my advisor was Dr. Rovahak, and he was leading the TCGA, the Cancer Genome Atlas Glioblastoma project. There, I had the opportunity to analyze real large-scale TB-sized genomic data sets. And then I gained skills to analyze different kinds of data sets like whole exome sequencing, whole genome sequencing, Bob-Conley sequencing, bisulfite sequencing, chip sequencing, ATAC sequencing, all sorts of different sequencing data. And then later I moved to Harvard as a senior bioinformatician. There I started to work on single-cell RNA sequencing and single-cell ATAC sequencing. 2 years later, I joined Dana-Farber Cancer Institute as a senior scientist first and a lead scientist to lead a bioinformatics team of 7 people to lead this big NIH-funded consortium project called CIDC, Cancer Immunological Data Commons. So there are actually 4 cancer centers in the States, MD Anderson Cancer Center, Dana-Farber, Mount Sinai, and Stanford. They carry out immunotherapy clinical trials in partnership with big pharma companies. And they profile those patients using NGS. And our team's responsibility was to process all those NGS data securely on the cloud, and then deliver them back to the clinical trial team for downstream qualitative analysis. And we are also responsible for some of the cross-trial data analysis. So stayed there for almost 2 years, and I joined my current company, Immunitas Therapeutics, and the Director of Computational Biology to establish the computational capability for the company. And Immunitas is a single-cell genomics company using single-cell RNA sequencing and single-cell TCR sequencing to find new targets for immuno-oncology. And our leading program, CD1621, is in clinical trial phase 1. We dosed several patients already, and it looks like it's safe, so it's exciting, and finger crossed that it does well for phase 2 and phase 3 and eventually can benefit more patients.
It's quite a journey across the Pacific Ocean and all over the US to various academic institutions and now into the private sector with a therapeutic solution in phase 1 trial. Why did you enter this field in the first place, and why have you continued on this journey in computational biology?
So first of all, for me, there are 2 reasons, right? To learn computational biology, usually there are 2 reasons for any people to do something. Like first is stay away from the pain. Second is you really like it, that's your passion. I think initially that was just the pain because I had to beg other people to do analysis for me, and definitely your project is not your priority. And I just said to myself, I had enough, let me just start to learn it myself. So initially that's just the motivation. But then later, after more and more like experience with computational biology, I really like it because I like the Unix commands. Initially, it could be intimidating, but after I master how to use Unix commands, I just couldn't go back to use the mouse and drag and click. and of course, for my current role, it's in a biotech company, and what we are doing now can really benefit patients and make a clinical impact. That's actually something I'm always passionate about. So if we can help others, save the patients, save lives, and that means a lot to me. Yeah.
What impact has computational biology had so far on immuno-oncology as a field? And what kind of work are you doing at Immunitas Therapeutics to advance understanding of immune responses in cancer or the development of therapeutic treatments to treat cancer?
Yeah, that's a great question. Because with the sequencing technology advancing so fast, the cost is just dropping so dramatically. But then again, the data is exploding. So I think last week I was in this conference, the Festival of Genomics and Biodata, and the sequencing cost that you will see, it dropped dramatically along the years. But then how to actually efficiently use those data, analyze data, and derive biological insights, that's still a very big challenge. And that's where computational biologists come to play a role to crack the code from those data. And for us, immuno-oncology, for any field, I think data is still a major block for us, because when you talk about data, you talk about the quality of the data or the quantity of the data. Because I did some wet lab stuff, so I know generating high quality data actually is not easy. So I really appreciate if the biology team for our company hand us really high quality data. I really appreciate that because it's a lot of work. And so quality of the data and the quantity of data, that means whether you have high throughput experiment assay in your company to generate those high throughput data. And of course, for us, we always use those public available data. But one of the major problems of those public available data is they're messy. It's inherently, that's just messy. And you need to spend a lot of effort to clean those metadata and put them together, harmonize them together in order for you to analyze or even just run any machine learning models on them.
So when you're coming into Immunitas and building this function of computational biology, where do you start? How do you grow that capability inside of a company like that?
Yeah, that's a great question. My perspective is that it really depends on the company that you are in, right? The big pharma companies is very different than the small biotech companies. As a small biotech company, you are always in the surviving mode. There are always deadline that you have to hit. So there should be a balance between perfect versus good enough. So for us, actually, when I joined, we had one computational biologist, and he established the computational platform using Terra, which was developed by Broad, and then also we use native Google Cloud, GCP, to do those large-scale single-cell data processing. And I would say, quick and dirty, initially, as long as it works, that's fine. Then gradually you can enhance different properties, make the process more streamlined and more reproducible, things like that. Of course, there are some other people think a little bit different. They think, okay, you have to be perfect even from the very beginning, but then you have to invest a lot of resources on that and to be perfect. But then you may actually miss the deadline because small biotech is just moving so fast. And that's actually my strategy, is move along, and as long as it's good enough to work and I don't break it,
Yeah, that makes a lot of sense. So you come in, are there already a set of priorities that Immunitas had in place for you? These are the questions that we want you to ask of our data? Or were you given the creative freedom to understand what was happening at the company and identify the priorities?
Yeah, initially I was recruited to do new target identification using machine learning approaches, still conventional machine learning approaches to find new targets. And at Immunitas, we can generate single-cell RNA sequencing data in-house, and also single-cell TCR sequencing data in-house, because we are an immuno-oncology company, and we're really interested in the T-cell phenotype. And we have this 10x genomics machine to do those single-cell profiling, and then we send those libraries to Broad for sequencing. So essentially, we grab all the data from the cloud and then process it in-house. And in terms of priority, at that time, we just want to find new targets. And the approach that we use is actually simple. For example, with 10x genomics, you can actually profile the gene expression level at single cell resolution, but you also get the TCR sequencing data from the same cell. So you can match them by the barcode. And that's going to give you a really actually powerful data sets, because now you can correlate whatever the gene expression pattern with the clonal expansion pattern. For example, the T cells, when they recognize tumor antigens or new antigens, they expand and then they replicate. So you can correlate the gene expression with that phenotype and then try to find which gene is positive correlated or negative correlated with that phenotype. And again, we're not using too fancy stuff here. And although we do use like deep learning for other purposes, let's just be honest, like, we don't need deep learning, and tell you the truth that we don't even need machine learning. So sometimes just simple statistics, like a correlation, will just tell you like which target to pursue. Yeah, sometimes. Yeah.
I love what you're saying about just using simple statistics, even to do something as complex as identifying drug targets from these correlated data sets. Are you giving formative input to the data that you want to generate from these next generation sequencing instruments, or are you collaborating with biologists in the wet lab who are having conversations with you about where the analysis path should go from here?
Yeah, exactly. So that's where my strengths actually plays, because I had the experience of doing wet lab stuff and I know their language. So I know how the experiment is carried out, and I had a clear question to ask. And Immunitas, actually, we sit actually together, like the wet biologist and the computational biologist, we sit next to each other. And when we have questions, we'll always ask them. And before they carry out any experiments, we'll discuss about the purpose of the experiment. What should we do? For example, we were really interested in target identification in the dendritic cells, which is a commander for T cells and NK cells. So it activates both T cells and NK cells. And for example, the public available data, they're very, because dendritic cells probably comprise of like maybe 1% of the total immune cells. And it's just very few if you don't enrich them. So for public data sets, you maybe find 10 cells like per sample. That's not enough for you to study. So then we can actually talk to the wet biologist in the company. Okay, we want to study this type of cells. If you do single cell RNA sequencing, can you please enrich it first? So they will do actually flow cytometry to enrich that subpopulation and send for sequence. Then we can actually get 1,000 cells per sample, which is a lot more than the public available data. But again, like computational biologist, in my perspective, is really to use computational skills to address biological questions. And you need to work really closely with the biologists. And when the data actually arrive, when we're doing those analyses, we just turn back and say, okay, we found this something interesting. Can you tell us this is like making sense or not? So we learn a lot of like knowledge from them. And we also explain those like complicated methods, for example, batch correction. What does that mean like to them? So we kind of learn from each other, and I really like that.
There are situations where you're picking up on something you think might be signal, and you're asking the wet lab biologist, what do you think of this? Could this be a signal of something important? And then maybe there's a data gathering exercise where you're figuring out how to amplify that signal so that you can validate what you've seen in the data. But then there's other situations where there are existing questions that someone has in the organization. And so you're collaborating to chase down whether or not the answers are there. Am I understanding that correctly?
Yeah, that's absolutely right. So we kind of learn from each other. Actually, sometimes biologically, probably that just a technical effect. The fact that if you just talk to them, we found this subset of cells that is interesting, then they'll tell you, okay, that's just probably when you process the samples, like maybe it's a heat shock, whatever. Like it's upregulated, it's purely like technical. So they can tell you that. And on the other hand, we can also tell them, for example, let me touch on this a little bit. For example, we are really interested in this gene called TLR9 and to try to quantify this gene's expression. And the thing is that, from the RNA sequencing data, like we find, why the gene expression level is so low and it should be high. And then when we dig into it, it really depends on what kind of computational tools you use for RNA sequencing quantification. And if you are aware, there are like those more conventional like methods, like you use star to align them and quantify them using HT-seq or like feature counts to quantify the number of reads that fall into a certain gene. But then for those methods, they cannot actually differentiate the reads that fall into exons that are overlapping from two different genes. For example, if you have overlapping exons, the reads that fall into those overlapping features, they will be discarded. And then you get reduced the number of counts. But we know, okay, the state of art, what other tools that we can use. And then we use this other tool called Salmon or Callisto. So those tools are alignment-free tools. So they are not checking those reads to the transcripts base by base. They're just checking whether it's compatible with the k-mers from the transcriptome model. And it's much faster. And it's also tends to be much more accurate. They will assign those reads that in the overlapping features that probabilistically to different genes. And then you rescue those counts. And of course, then they also learn that from us. So it's like mutual learning process.
Yeah, I love that example.
Are you a biotechnology company looking to unlock the potential of your business data? CorrDyn can help. We're an enterprise data specialist that helps companies working in life sciences make smarter strategic decisions. From developing the right data strategy that starts with our data maturity assessment to building and delivering bespoke technical solutions, we are equipped to tackle the most complex data challenges. We have partnered with dozens of high-growth organizations, from manufacturers of custom oligonucleotides to molecular diagnostic companies, to achieve data competency. Whether you need to supplement existing technology teams with specialist expertise or launch a data program that lays the groundwork for future internal hires, you can partner with CorrDyn to unlock the potential of your business data today. Simply visit connect.corrDyn.com/biotech to learn more. Now, back to the show.
Is there a typical day for you in your role as director of computational biology, or is it just every day you're coming in and building on the work you did the previous day, like answering the new questions of the moment?
Yeah, that's a good question. I think as a manager, so first of all, I had to actually set up a goal for the year. So I think there's a long-term goal. So usually we have a document to write down, okay, what we need to accomplish this year. So you need to have one or two big things that you want to accomplish in this year. Yeah. And then you also set quarterly goals and then weekly goals, and you assign those tasks to your team members. So my typical day as a small biotech, even as I'm a manager, I still do a lot of hands-on analysis. But again, at the same time, I also need to supervise 3 members before, and then my job as a manager is also to develop them, because I want them to be successful and their success is my success. So I also learned those management skills and also leadership skills along the way. And even when I was at Dana-Farber, so I took leadership classes and some coaching sessions, and to learn the responsibility of a manager is really to help your direct reports to be their best version. And I fully agree with that. And that's what I practice. Yeah.
That makes a lot of sense. Can you give some examples of, or a template for what an annual goal looks like in your computational biology role?
For example, when I just joined the company, maybe one of the goals is that, okay, let's take all the public available data and harmonize them and establish a pipeline that we can process them uniformly in-house. So that's what we did. So then actually wrote SNCMake pipeline just to process all this data uniformly on the cloud. And also we developed this tool to annotate those cell types uniformly. So then we can have the same cell type that is annotated for different data sets, because that's essentially what you want to do. Okay, across different data sets of different cancer types, let's check the gene expression of a particular gene across different cancer types, but in a specific cell type. But then if you don't harmonize the cell type label across different data sets, you can't do that. So that's one of the major goals for a year.
Right. So it's setting up the scalable data infrastructure that's going to enable you to do the kinds of repeatably and efficiently over time.
Correct. And of course, if you ask me again today, I don't know, I might actually just turn to some other vendors. Again, it really depends on your strategy. If you want to be quick, and then you can also spend some money just working with vendors. I know vendors such as like Elucidata. They actually harmonize all the single cell data for you, and then you can just request. Another platform will be BioTuring. Like they are really good at single cell RNA sequencing data curation. Also, they expanded to like spatial transcriptome as well. And high quality data and harmonize all this metadata using ontologies, so you can easily search. But we still need to have some in-house capability, because if you are relying on them, they process data takes time. Right? So if there's a newly public data set, you want to do it, and then we request it, then it will take some time for them to do it for you. So that's why we still have some in-house capability. Okay, we can crunch the data ourselves when needed.
Interesting. I know that you're monitoring the research as it's come out. To anyone who doesn't follow you on LinkedIn, I would recommend following you on LinkedIn because you share a lot of papers. So are you keeping an eye out for new data sets that get published that you can then pull into your pipeline and combine with the infrastructure that you've already got?
Yeah, that's exactly like why I'm on like social media. So one of the main reasons is that I follow like most recent papers, bioinformatics tools, and then I'm aware like what data sets could be available, what data sets are relevant, especially for single cell immuno-oncology. I will just write it down. We have an Excel spreadsheet. You always start with the Excel spreadsheet. So then I will put the title of the paper and also the link of the data there. So in case we want to use it, then we can actually process that by ourselves. And I also share a lot of tools like bioinformatics tools, like every day, especially for single cell genomics. You probably see tools every day published. It's just too much and it can be overwhelming. And I read the abstracts a little bit, and then I'll know this could be useful for other people, and I will retweet or post it on LinkedIn, but I will also curate them in my GitHub repository. So that's actually my superpower. If I write it down, when I need to use something and when I had a problem, I can somehow remember and I can quickly find that link and okay, let's try that tool. And of course, before I try that tool, I will take a look at the documentation. So no matter how good your tool is, if the documentation is not good, so I'm not going to use it because I don't know how to use it.
Yeah. I'm interested in your ideas about the tool proliferation that we see. In what ways is that enabling you to do new things that you've never been able to do before? And in what ways is it confusing people in the field and making it harder to do the kind of work that you do?
It's a double-edged sword, right? Like it's on the one hand, there's so many tools published, and for single cell specifically, and you really need to be able to pick one good one, right? But then without testing it, it's really hard to know. And I think that's one of the problems, probably. You just don't know which one is good. And as you said, it's probably too fragmented. And in terms of data analysis, even for single cell, I know it's from 10x Genomics, like most of the people use their platform, and they have their cell ranger, whatever software to do those pre-processing. And some other people use like Callisto or Salmon, they also have a single cell version to process those data. And it turns out for me, like we were actually using a quite old like Widow WDL like workflow on Terra, and it was using a really old Cell Ranger version, and it's like 3.1 something like that. And then I tried this like Callisto or like Salmon. It actually can detect much more like genes and much more transcripts per cell, and then also detect more cells, and it can recover more cells from your experiment. And again, different people use different tools, and then the count table eventually like people get from different methods. It's really hard to do a meta-analysis, for example, like how do you actually harmonize them if they're from different process pipeline? Or what you can do, you can start from the raw FASTQs and process them all at the same time, but then that also takes like a computing resource, and that can be a lot of work.
Those are some interesting challenges. You mentioned that you're in Google Cloud, and I'm just interested, like, how do you use the cloud to enable the workflows that you're talking about? Like, where does the cloud come into play?
Yeah, the good thing about cloud is that it's so scalable because you can use as many CPUs as you want and as big as disk that you want. And I think for single cell data, the challenge is that data can be big. For example, some data sets can have like millions of cells. It's just hard to do that on my local desktop. And for pre-processing, again, I mentioned we use this snake make pipeline, so we can, and it's also Dockerized, partly for the reproducibility purpose, and it's easy to run. So we wrap it in a Docker container and just run commands, and with the same actually input format, we can actually crunch the data regularly and without too much human intervention. So that helps to improve our reproducibility. And the other thing is also about data storage, right? On the cloud, you have a lot big space, then you just need to pay for the storage. Just be careful, it can actually accumulate so fast because the raw FASTQ files is so big, and make sure you put them in the cold storage so it's much cheaper. And again, you have to actually really organize how you actually store for example, the raw input, pipeline output, and also the standardized whatever the downstream analysis output. So you have to actually organize all those infrastructures.
That makes a lot of sense. You have these raw files that you're storing in a cloud storage environment, and then you have these pre-processing pipelines that are Dockerized that you're deploying whatever in a virtual machine or in Cloud Run or something like that. Do you use a cloud data warehouse like BigQuery at any point in that process, or are so many of the analyses that you're doing using file systems and Docker containers that there's no room for that sort of thing?
Yeah, we haven't used any of those like big data warehouse stuff yet. And I think it really depends on what type of company you are in. If your company is generating like tons of data like in-house, then it can be beneficial to have such a thing that you can easily query the data. And for us, I think one of the major challenges actually is metadata tracking. I think how do you track those metadata? Even for public data, you have to actually clean them, actually go to the supplementary information tables and go to geo, like they may be hidden in some like places in the geo record. And it's just a lot of work. And then the spreadsheet is also not greatly formatted. Like if you read into like for the Python or R, then you have to spend some time to clean it up. And different data sets, they may also have different metadata columns. And even they have the same column meaning the same thing, they may be named differently. And also the content of the column may be also different. For example, female, like it could be F or capital female, lowercase female. So you need to actually harmonize all those stuff. Actually, that's one of the major hurdle, I think, for machine learning models or any even data science and to the models to work on those data.
So for the metadata, do you store the metadata in a database or something like that and then reference the metadata as you're pulling the raw data through the files, or do you just write the metadata back to files that you're storing in cloud storage?
Yeah, that's a great question. So now we just have like spreadsheet. Like when you have like small enough data sets, like if you still have less than like 1,000, I would say like a spreadsheet still works. So again, I think perfect is the enemy of good. So, but if you are generating like again high throughput data every day, like easily explode. I believe like even Broad, when they first started like the CCLE, the cancer cell line project, they also started with a spreadsheet. Then I heard that until one day they couldn't handle it. Okay, let's do something more complicated. Yeah,
that makes a lot of sense. So you mentioned machine learning, and you've talked about situations where you need machine learning and the kinds of analysis that you're doing, and the situations where a statistical test or a data visualization is good enough to tell you exactly what you need to know. How do you determine where machine learning should best play in the kinds of computational biology approaches you're using?
Yeah, that's a great question. As far as I can see, like I always try like simple machine learning model first. And for example, in this TCR versus gene expression example, like we just use conventional methods that for that purpose, like random forests usually perform well, and even logistic regression is really interpretable, and you can get the weights of the features and whether it's positive or negatively correlated from the logistic regression. But then for other like more complicated data analysis, for example, the image analysis, that's where I think deep learning is shine, because I think those convolutional network, they really do well like on image processing. And you may also be aware of AlphaFold. They can predict protein structures really well. I think in those cases where deep learning can benefit the computational biology. And we actually did try deep learning using AlphaFold to to, for example, predict a potential like a ligand for a target that we are interested in. Of course, we get some like top hits, and we ask the wet biology team to verify, and it didn't work. But again, like we tried. So I think it's still not there. And also recently there are several, I think, drugs they are on clinical trial, which are designed by AI, like generative AI, like small molecules, and also like large molecules, like antibody sequences. And they are still in early phase, like phase 1, phase 2, and we still need time to see how they perform, like in the later trials, whether they can hit the market. But definitely when we have enough data, definitely deep learning or machine learning approaches can have a really great potential.
So I haven't heard you talked about a lot of images in your data pipeline today at Immunitas. Is that something that's on the horizon for you, or is that something that you're already working with?
We don't work on a lot of like image data, but recently we purchased this spatial transcriptome platform called VisGene. You can look at a single cell gene expression level on the tissue slice, but only you can look at 500 some genes. And it's an image-based platform. So again, for that, you have to do like cell segmentation and then assign the transcripts to each cell. And we're doing a little bit on that, but we're using like existing algorithms like CellPose, and it does a pretty decent job at this moment. So we are not actually working on image processing per se, but we touch a little bit on that. Yeah.
Interesting. I really appreciate the pragmatic approach that you use, where you're like using the simplest, most understandable methods at any point in the process, and you only move to the more complicated method when you feel like the question necessitates the application of that more complicated method.
Yeah, exactly. That's why I think the AI models, like people, I think there's really hype there. Okay, we can address this using AI. We can use that. We can address that use AI. I think we still have a long way to go, and really don't overcomplicate the problems. Like if it can be answered using a simple methods, and then just use a simple methods, and it's more interpretable, I would say, with the simple methods. And with the more complicated method, you don't really know, especially for deep learning models, it's a black box, like how many layers, how many units that you need to have for each layer. And there's so many hyperparameters you have to tune, and it just doesn't work.
Interesting. Given that AI is not there yet, as you look out 1 year or 2 years from now, where do you see the biggest opportunity for computational biology to really contribute to immuno-oncology? Is it just application of the same methods but with better data? Or are there methodological improvements that you're going to see? And if so, just any insight you can share there.
Yeah, I still think the data is the key. And if you have a better data, high quality data, more data, and your machine learning models that can be trained on those data, then you may have some sensible predictions. And that's the thing, just like Google or like any other, like those big companies, you'll see who will win actually in the AI war, like for those fintech companies. And I would say Google, because Google has the most data. For example, YouTube, all the data on YouTube, they can transcribe them into like words, and then they can train their whatever large language model on those. It's huge data sets. And like ChatGPT, it's really popular, and OpenAI has pioneered that. And again, if they don't have data, that's their biggest disadvantage, because if you don't have data, that you don't have the moat, because there are many also like open source large language models. They can even perform as well as the OpenAI model. So the model itself, I would say, will become a commodity. But then the data really is the key, that is your moat, and then you can really apply your machine learning approaches on those data. And that's why you really need to have a platform that can generate those high throughput data in-house. And that's what I'm thinking about. That's platform first, AI on top. So you can generate data, then you can apply your models on those data. And that can potentially give you some good, for example, new targets and even like new molecules, drugs, like protein sequences, like de novo protein engineer and antibody engineer, that can potentially give you some sensible results and can speed up the whole process of drug discovery and development.
So when I think about data as a competitive advantage at a company like Immunitas, there's the quality of the questions that you're asking and the collaboration between the computational biologist and the wet lab biologist. And then there's the quality of the data that you can generate and the volume of data that you can generate that provides insight into those questions. And those are the sorts of places where you can really gain an advantage. And then the methods that you use to analyze those data are relatively commoditized. You just need to have sort of the capacity in-house to do that kind of analysis. Is that right?
Yeah, I want to echo your first point. Like, really, you need to ask the right question. So that's actually even more important than your model, than your data, because if you ask the right question, you may not need a huge data sets. You just need the right specific data sets. And if they are not available, then we can design an experiment and generate their data sets.
Mm-hmm.
I think I really appreciate that point.
As a practice, when you're growing a computational biology team, I imagine that one of your roles as a manager is to like help your people to ask better questions. How do you help them to do that?
I usually sit with them and to draft out their career development plan. So what they want to learn. And for example, if you want to learn a little bit more about biology, immunology, I will encourage them to take courses on Coursera, edX, those open platforms. And if they want to learn more machine learning, deep learning, I encourage them to take courses as well. And I also bought those books, machine learning books. And by the way, this one really good if George Stama, he has this really nice machine learning books, use those cartoons and use plain language to explain those complicated models. And I find it's really useful. And I bought several copies to them, and also bought several just more practical coding books for them. So it's just a learning process, and I actually learn together with them. So you have to foster a really good learning environment together as a group, and then we share different expertise with each other. And we have this Friday teach and learn session. So each time, one people will present one thing that they know well, and maybe other people don't know, so we can learn from each other. And so I also encourage them to think about questions proactively, how to think in the big picture. So not only technical things that I can write this code to analyze this data, but before you even analyze data, what kind of question are you asking? Is this data set that is suitable for answering that question? That's actually more important. Yeah.
I heard a couple of things in there. At an individual level, you want people to have the full scope of capabilities that they need as a computational biologist. So you want people to have the biological grounding to be able to understand what's happening in the data and in the wet lab. You want people to have the statistical and technical grounding to be able to understand how the data sets are structured and how to clean them, and then also the methods that they apply when they're analyzing that data.
Yes, correct.
Interesting. And then the other thing I heard was the culture that you foster as a team, where if all of you are going down this journey together, then you're all going to learn much faster than if one of you is just consuming resources all day on archive, for example.
Yeah, I really appreciate this saying. If you want to go fast, go alone. But if you want to go far, go together. So you can always learn much more if you learn together.
Yeah, so I've noticed that you share a lot of resources in public about bioinformatics, about computational biology. What motivates you to share and build community around computational biology?
Yeah, I think initially I didn't think too much. I was just posting, and sometimes I was posting just complaints or about bioinformatics or how difficult this problem is. But then gradually I started to post the solutions that I have, or the new tools that I found that it could be useful for other people. And because I went through the pain, I didn't know where to start 10 years ago. Although I was speaking my transition from a web biology to a computational biology for 10 minutes or so, but it really took me 10 years of grinding and learning, relearning, and frustration, and I didn't know how to start. So one of the purposes is that I want to help others to avoid the same pain. So at least when they started, if they decide to make the switch or they want to learn, they know where to start, and they have a guidelines, what course they can take, what books they can read. So I have some posts about the books that you can use, the courses that you can take. And I think 10 years after I started, there are much more resources there. And I also helped you to curate them, and it's much easier for you to start now today. So I think the one of the purpose that I started to share is to to help others to avoid the same pain.
Yeah. Just at a high level, as you think about your journey and all the resources that you share and the sort of people that you manage, for people who are interested in getting deeper into computational biology, what would you recommend to them as a starting point?
Starting point. So I have a lot of like resources on my GitHub repository and also my own website. Blog posts. I write many blog posts to explain like some technical stuff, and sometimes it's more like general. Talk about AI, like in general, has it revolutionized drug discovery or not? So I write all sorts of things. So you can go there and take a look. But I think in one of the GitHub repository, I listed all the courses that you can take. Make sure you go there and then you can take a look so you can follow the guideline there.
Yeah, that sounds good. And we'll make sure to include some of those links in the show notes and in the blog posts that we write about this episode. As we bring the conversation to a close, I'm just interested, are there any resources that you've discovered recently or ideas that have come about recently that are on your mind that you feel like are at the leading edge?
Yeah, you were like asking what kind of platforms we were using, and I told you Google Cloud. And to be honest, establishing a Google Cloud platform, it can be like painful as a small biotech because you have to actually deal with all this. So to set up a GCP, you have to set up organization and set up the administration, like security, all sorts of things. And recently, I had this problem. Okay, when I attach a disk to my computing engine, and then it turns out that disk is too small. And okay, how can I actually just pause the engine and increase the disk? Then I have to Google around and find the menu and do that. There's some commands that you can type. It can be actually challenging also sometimes. So recently there are like other platforms that are emerging. Of course, before that, there are like old platforms like DNA Nexus, Seven Bridges. Those are old companies. And there's some new companies, for example, Watershed, Ledge Bio, and even Deep Origin. Those type of platforms, they are trying to actually solve these pain points for bioinformaticians. So they have their platform ready. So you don't have to set up all those like GCP stuff or Amazon cloud computing. You can just, okay, I want 6 CPUs. I want this big machine. You can just like easily select them, and then we'll handle all the security, all the stuff behind the thing. And it's just much more actually convenient for us and save us time. So I actually look forward to try some of them like in the future.
Yeah, so these are like cloud computing platforms that are designed specifically for the workflow of the computational biologist. Obviously, I've experienced the pain of being in different cloud environments and being challenged to figure out how to accomplish a particular task. And there's a lot of documentation and a lot of tools on the table. And it's very easy to get confused. So I'm interested in what are the aspects of a platform like that you feel would be most valuable to you in moving from one cloud to the other?
So I think one of the main thing that I care about is reproducible computing. I'm an advocate for reproducible research, and how can you actually integrate that into the platform? It's important. For example, now we're using Docker container, but that also like Git version control for the code. But then again, you also, for those files that are generated by the pipeline, you also need to version control the data. So if a platform can like help to advance all those aspects and the computing environment, the code, and also the data, version control, all three of them, that could be really helpful. For example, you generate the data, you really know, okay, there's probably a unique ID that is attached for that data. So you can directly trace back, okay, how I generate that data, what command did I use, when did I generate that data? And that could be really helpful, yeah.
Yeah, each time you rerun that pipeline, it can be considered like an experiment, and you want to track the different metadata surrounding that experiment, including the methods that were applied, the compute that was used, the specific data resources that were inputs, the specific data resources that were outputs, and be able to return to them later on and look at differences between them over time.
Yeah, exactly. And for now, like what we are doing at this moment practically is that, okay, let's run our stuff on Docker container, Git version control them on GitHub. And when you finish this analysis, make a deck, and then put in the last slide, put your notebook link into the last slide. Then you can trace it back how you generated data, like in a not perfect way, but it's good enough for this moment.
Yeah, but As your team grows, as you're doing more work for a larger organization, the complexity and the number of experiments that you're running as a computational biology team is growing, and you want to be able to navigate that data through a single interface. Yeah, that's really interesting. Tommy, this has been really insightful. I really appreciate you coming on and look forward to connecting down the line.
Thank you very much, Ross. And I really enjoyed the chat.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.